This is DNA Structure and Replication Modeling Project by Polina and Livia. This is a DNA nucleotide made up of phosphate groups, deoxyribose sugars, and nitrogenous bases. Topoisomerase is an enzyme at the head of the replication fork, and its function is to prevent supercoiling. Adenine, thymine, cytosine, and guanine are connected through hydrogen bonds. The gummy bears represent the nitrogenous bases, and the toothpicks represent the hydrogen bonds. Replicase is an enzyme that breaks up hydrogen bonds between nitrogenous bases, and single-strand binding proteins coat DNA around the replication fork to prevent rewinding of DNA. In the leading strand, primase synthesizes RNA primers complementary to the DNA strand. After this, the polymerase extends the primers. This adds free nucleotides to the 3' end. This makes the bulk of the DNA. An enzyme called ligase seals the gap of the DNA fragments. Ligase is here represented by the Jolly Rancher chews, which are on the red Twizzler. The lighter pink Jolly Rancher chews are a DNA polymerase, which replicate DNA in the 5' to 3' direction. The new strand created is also in the 5' to 3' direction. This means that only one DNA segment is required in the leading strand. The lagging strand first synthesizes RNA primers. DNA polymerase III extends the primers, then DNA polymerase I removes and replaces the RNA primers. DNA fragments, called Okazaki fragments, are built because the lagging strand goes in a different direction, which are then sealed by an enzyme ligase.